Gene therapy, Vivo versus Vitro, Vivo to Vitro. In ex vivo gene therapy, as we show here, current vector with therapeutic gene combined with host cells in vitro in the lab. After that, after making the genetic engineering for the host cells, the engineered cells containing here therapeutic gene. Now, the ready engineering cells containing therapeutic gene will be injected in the femur of the patient suffering from what? From defective gene. Why? In vivo, the targetable injectable factor will be introduced in the arm of the patients as a vivo or as in vivo gene therapy. That is represents ex vivo gene therapy in addition to in vivo gene therapy. Vivo to vitro gene therapy. What gene therapy can achieve? Replacing a mutated gene that causes disease with a healthy copy of gene and activating or knocking out mutated gene that is functioning improperly and introducing a new gene into the body to help fight disease. Now, genes are necessary. It's very important thought here. Gene therapy is the use of gene as medicine. Is the use of genes as medicine. It involves the transfer of a therapeutic or working gene copy into specific cell of an individual in order to repair a faulty gene copy. Thus, it may be used to replace a faulty gene or to introduce a new gene whose function is to cure or to further modify the clinical course of condition. The steps in gene therapy is being graduated from DNA to gene encapsulated in adenovirus here and in vivo gene therapy in the arm of patients. Adenovirus releases gene to the cells and the target cells here will be target cells here will be containing here therapeutic gene. After that, the therapeutic gene will giving a functional protein instead of dysfunctional protein. And instead of dysfunctional protein. The goal here is how to replace dysfunctional protein, how to stop the continuous release of dysfunctional protein and creation a functional protein. Here, functional protein coded by functional gene will give a healthy property, healthy characters of the life. Yeah. Okay. Now, here in this schematic representation, how viral factor particles are produced by engineered producer cells. After producer cells, here viral gene therapy and overview. Producer cells, here factor particles carry therapeutic gene and deliver them to the target cells. The therapeutic protein may act inside or outside the cells, and in some applications, these components of insert the gene into the chromosome of the target cells, as you show here. Yeah. What are the manipulation correct? How manipulation correct the defective genes? How correction will occur? Now, as you show here, sorry, in vitro manipulation, this represents the dysfunctional gene. And this functional gene, this functional gene will be self-replication. Self-replication to corrected cells. Here, the functional or therapeutic gene will be introduced neighboring to what? To this functional gene. Here, each cell containing dysfunctional and functional gene. This functional is labeled by green color here, while the functional is encoded by red color here. Okay, the corrected cell now will be proliferated, and viral vector will be released, and here is a viral vector therapeutic gene at other meaning. Viral vector or therapeutic gene will be introduced as therapeutic gene neighboring to the dysfunctional gene, and here the corrected cells. Gene therapy, I repeat this very important sentence, this therapy is the insertion or introduction the therapeutic gene instead of none of its functional gene to create functional protein that provide me the healthy characteristics. Now, after we will understand gene therapy, now what does it mean by antisense therapy? Till now, do you have any questions regarding what I said here? Okay. Now, what does it mean by antisense therapy? Antisense therapy is a form of treatment for genetic disorder or infection. When the genetic sequence of a particular gene is known to be causative of particular disease, it's possible to synthesize a strain of nucleic acid, DNA, or RNA, or chemical analog that will be bound to the messenger RNA produced by the gene and inactivate it effectively, turning the gene off, as you show here. Now, the normal process is the mRNA will be migrated in normal process of protein synthesis. The mRNA will be transferred from the nucleus to the cytoplasm, and here the mRNA as anticodon, as codon anticodon here, and will be going to the ribosome, to the site of protein synthesis. In antisense therapy, the certain sequences will be bound to the antisense here and stopping the passage of messenger RNA to be going to the what? To the ribosome to synthesize the protein. In this stage, the process will be stopped. Therapeutic sequences will bind to the antisense here, will bind to the codon with the sequences on the tRNA and prevent mRNA from going to the ribosome to synthesize the protein. Yeah. Antisense therapy is not strictly a form of gene therapy. It's not a form of gene therapy, but it's genetically mediated therapy and is often considered together with other methods. Okay. Now, gene therapy depends on the delivery of correct genes. Viral factors. Viral factors are tools commonly used by molecular biologists to deliver genetic material into cells. This process can be performed inside a living organism in vivo or in cell culture in vitro. Viruses have evolved specialized molecular mechanisms to efficiently transport the genome inside the cell and then infect. Viruses are used as delivery tools. Viruses are used as vector to introduce the genetic material inside the bodies. These viruses are inactivated. They are not able to reproduce. They are not able to reproduce. It's genetically engineered. Like adenovirus, virus, that's one from DNA tumor virus. Rotavirus is one of RNA tumor viruses. Somatic and germ-line therapy. Now, in the world, the researchers will be going now to somatic and germ-line gene therapy. In gene therapy can target somatic body or germ, egg, and sperm cells. In somatic gene therapy, the patient's genome is changed, but the change is not passed to the next generation. Whereas the germ-line gene therapy, the newly introduced gene is passed on to the offspring. Now, the world is mainly thought of how to create the 42nd chromosome. Researchers are also experimental with introducing a 42nd artificial chromosome to the body. It would exist autonomously alongside of the other 46, not overlaying, not affecting their working or causing any mutations. It would be large factor capable of carrying substantial amounts of genetic information, and the body's immune system would not attack it. Will interfere in gene therapy, and this characteristic model, how low would be interferes in gene therapy. So that is enough. If you have any question, I am ready to answer. Yes? Yeah, the gene therapy is used as medicine now. The gene are used as medicine. I think with the passage of time, this line of treatment will be going to be in the phase of the list of therapy. I think that. Okay. Thank you very much for listening to my lecture. Thanks. Thank you.